So today we are going to see a theory portions on molecular biology. So this we will be uploading in several parts. So please watch our channel, subscribe our channel, like our channel, and also put comment in the comment box. Press bell icon to receive notifications on new videos what we are uploading. So what we are so we are very familiar with the term molecular biology, right? So nowadays there is nobody who don't know or who never had a term like molecular biology. So all students, PhD students, research students, all are doing genetic engineering work or should know molecular biology very well to do the experiments and to understand the concept properly or to devise a new experimental setup or experimental process. So this molecular biology term was first coined by Warren Weaver. Warren Weaver, when he was preparing a proposal or documentation for Rockefeller Foundation, he proposed this term in 1938. Molecular biology, he coined this term. So the people were that time very well versed with the technologies like biochemistry, genetics, and physics like that. So these were all independent subjects. There was no intercorrelation or interconnection between these subjects. So when they were trying to address a biological function or a biological process, they were either doing a biochemistry as a discipline or they were following genetics as a discipline or physics, chemistry, crystallography, genetics, immunology, developmental biology, like that. So it was a interdisciplinary subject. So interdisciplinary. So many different disciplines were handling all these things. So when they try to put it all together to understand the biological function, they want to coin a new term, and that is how this new discipline, molecular biology, was evolved. Okay, so now we all discuss molecular biology. So how this molecular biology evolved? So there were two persons. One who that is Gregor Mendel, and Frederick Miescher. So Gregor Mendel and Frederick Miescher, so they contributed two concepts for the evolvement of molecular biology. So Gregor Mendel, we know, he was an Austrian monk and botanist, and he proposed laws on inheritance. Laws on inheritance, and he proposed three laws, that is law of segregation, law of independent assortment, and law of dominance. So he was having pea plants in his garden, and he was observing them, and he found that certain characters are how it is transmitted to the next generation, like that. He was observing it. So he took certain characters of that plant, and he was able to see that certain characters are inheriting from its parents to offspring in a specific manner. So I am not going to detail into of these each laws, because the, people will be already knowing all these things in the subject genetics. So in the law of segregation, what he is saying is, suppose if you take a flower color, so that flower color will be having two alleles. Okay, so that two alleles will be there, and one will be, or you can say, if there is a pea plant is having red color and white color. For red color there is one allele is there, and for white color there is one allele is there. And when the gamete is forming in the plant, these two are segregating, and that is how this character is come. You can also see it in the offspring. So that is the law of segregation. And in the second one, law of independent assortment, what he is saying is, there is two independent characters. You say one is the pod shape, another is flower color. So the flower color, there will be a red color will be there, and there will be a white color will be there. And for pod, there will be non-wrinkled will be there, wrinkled will be there. So all these things are not are independently assorting in the gamete formation. That is how you can see different combinations of these characters in the offspring. So that is how it is called a law of independent assortment. And the law of dominance, what he is saying is, suppose when you take the flower color, red color and white color, and red color is always dominant than the white color. So like that, he was able to see certain characters which are present in the pea plant and how it is getting transmitted to the offspring. So that is how he proposed all these three laws. And so this was a first one. So he proposed these laws very earlier, and till 9900, nobody was looking into this. And later only people looked into these laws and tried to understand what he written in the document, like that. So there was another person, that is Frederick Miescher. So Frederick Miescher also, he was a not a molecular biologist or biochemist or physicist. He was a physician. So he was handling wounds from patients. He was handling bandages of that. So he saw pus present on these bandages. So because of curiosity, he started treating this pus which was present on the bandages with the protease enzyme. And he found that he obtained a particular fraction which is rich in phosphorus. So which is rich in phosphorus, and he called that as nuclei. And he saw same thing in when he was doing digestion on the sperm cells also. So he later what he did is his student Richard Altman. What he did is he further did studies on this nuclein, and he found that it is a mixture of protein and a phosphorus rich material. Frederick Miescher also found that this nuclein is having an acidic material, and Richard Altman, he separated that. So he found the protein is there and phosphorus is there, and this phosphorus is a rich compound, is actually the acidic one. So because it is isolated from nuclein, he named it as nucleic acid. So described it as nucleic acid. So the experiments by Mendel and the experiments by Altman, it was independently done, and people for a long time they not looked into it uh, seriously. And Richard Altman also was thinking that this nucleic acid is a storehouse of phosphorus in the cell. They never thought it is a trait, or it is the trait what it was described by Gregor Mendel. So it was all independently written. So it was all unconnected. People are not able to connect it all together like that. So the Mendel's experiment, he called those characters as traits. So it said that there are some traits which are present, and these traits are responsible for the color or code characters, etc., like that. So the flower color or the pod shape, what they are saying, they called it as phenotype, like that. So they were not able to correlate the alleles or traits with the phenotype or like that, or you can say they were not able to correlate a genotype and phenotype of the organism. So that time there was another physics, not physicist, physician. His name is Archibald Garrod. Archibald Garrod. He was actually handling patients suffering from alkaptonuria. So alkaptonuria is a condition where the patient will be having a very dark colored urine. So this dark color is due to homogenesic acid. So the dark color of the urine is due to homogenesic acid. So this is actually a breakdown product from tyrosine, amino acid tyrosine. So to convert the homogenesic acid into a colorless compound, there is an enzyme present, and that enzyme is absent in the alkaptonuria patient. So he found that that particular gene is recessive, and some persons do not have that enzyme. So because of that, their urine color will be dark. So during that time also, it was not able to connect with the genotype. So it was later two scientists like George Biddle and Edward Tatum. So these two persons, scientists, were working on a mold called a Neurospora crassa. Neurospora crassa. It is a mold actually. So what they were doing is they were experimenting with this, and they were irradiating with X-rays and UV rays. So they irradiated this Neurospora crassa with X-rays and UV rays, and they found that they got certain colonies, and they have to supplement that colonies for growth, and they named them as auxotrophs. So the Neurospora crassa, originally what they have used is was not requiring any supplements for growth. But after X-ray radiation and or UV radiation, what they found is they got a certain number of colonies which require some supplement to grow, either vitamins or amino acid or some other carbon sources, something like that. And they found and they named them as auxotrope because only when a supplement is given, it is growing. And then they found that these X-rays and UV rays are causing damage in the DNA. So when there is a damage is introduced, there is a change in the phenotype. And they were also able to see a observation like how the Archibald Garrod was observed. Means homogenic acid is accumulating because there is a downstream enzyme is absent. So like that, they have found that there are some downstream enzyme is absent. Because of that, its predecessor is accumulating in the cell, or it requires some other supplement for growth because some enzyme is absent for that production of that particular compound like that. So that is how they found that there is a particular portion in the DNA which is actually determining a particular enzyme, and they proposed it as one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. And they found that this one enzyme, what it means is not made by a single polypeptide. Some case, one enzyme will have more than one polypeptide. So because of that, they again it was reformulated like one gene, one polypeptide hypothesis. Like that. So that is how they found that there is a correlation is existing between the DNA and the phenotype what we are observing. So in the next video, I will tell you how people coined the term DNA, how they coined gene like that, and what is the composition of DNA, and how a DNA you can say a gene like that. So thank you for listening. So we'll be uploading the concepts in molecular biology in different parts. So keep watching. Thank you very much.